Hello, folks. In this video, I want to talk to you about the autonomic nervous system or the visceral nervous system and the two major branches of the autonomic nervous system. That's the sympathetic and the parasympathetic. These are separated both by anatomy as well as the effect that it has on the body's alertness and ability to do intense activity. So we're going to look at the organization. We'll contrast these two nervous systems in terms of organization and what the effects that they have. And in doing that, we need to look at their synapses, the neurotransmitters they use, and the types of receptors that they use. So you'll remember this from long ago, the two divisions of the nervous system compared. We have our somatic portion of the nervous system here and autonomic or visceral here. And remember our somatic, this is just looking at the motor activity, right? And that's what we're going to really focus on here is motor activity. The motor neurons have their origin in the motor cortex of the cerebrum. So in this part of our brain that is doing the conscious planning and thinking and that sort of thing. So conscious motor output, right? And you'll see that there's two motor neurons there in this pathway, this first order and second order neuron, and that the effectors are always skeletal muscle. Okay. In contrast to the autonomic or visceral nervous system, where they have their first order motor neurons in the mid and the another part of the diencephalon, not this huge cerebrum or conscious part of the nervous system where we're doing all our planning, but in the thalamus and hypothalamus, and primarily in the hypothalamus of the diencephalon. And you'll notice too that these have three neurons, a first, second, and third order motor neuron. And importantly, that all their effectors are not, they're everything but skeletal muscle, right? So smooth muscle, like the muscle surrounding your arteries and veins, and the muscle surrounding your digestive tube, and the muscle of surrounding your airway, right? Glands, right? Like salivary glands or gastric glands that are secreting digestive enzymes and acids into your stomach, or even sweat glands. Cardiac muscle, this is just heart muscle, so and only heart muscle. So the muscles of your heart are innervated by these autonomic neurons and adipocytes or fat cells. So this is the last type of effector. So if we're innervating fat cells, we know we're going to see increases or decreases in fat metabolism, the storage of fat or breakdown of fat and liberation into the bloodstream. So these are very different effectors than we have in our somatic nervous system. Okay, so let's think about now the two divisions of the autonomic or visceral nervous system. There's the sympathetic division and there's the parasympathetic division. So the sympathetic division, and when we talk about the sympathetic portion of the autonomic nervous system being stimulated, this is the part of the nervous system that prepares us for intense activity. It's often called the fight. I reversed those fight or flight division of the nervous system. It prepares us uh, to run away or to fight. And that means a bunch of things. We need to be alert and attentive to what's going on in our surroundings. We need to have the ability to deliver the nutrients that we need to feed our skeletal muscles, because those are, of course, essential in both of those activities, fighting or fleeing. Um, and we need to put off sort of general maintenance activities like digesting food and urinating or defecating until a later time, right? So when we think about sympathetic stimulation and the sympathetic portion of the nervous system, this is what we tend to get. We get increases in mental alertness. So we have their effects on the central nervous system. There are lots of effects then on peripheral effectors, both some skeletal muscle and all of these sweat glands and digestive glands and smooth muscle and that sort of thing. So we get like contraction of the ciliary muscles, and we'll learn more about the ciliary muscles in the special senses, but they're basically a muscle within your eye that controls how thick your lens is and tends to bend incoming light and focus it on the photosensitive part of your eye, which is your retina. So this helps us to focus on objects that are close to us. And it also affects smooth muscle of the iris, which is the colored part of your eye, and it causes a certain muscle in there, the radial muscles, to contract. And what that does is to basically shrink the colored part of your eye and increase the pupil diameter. This allows more light into the eye and allows us to be, even in low light situations, to create a clear picture of what's in front of us. It also results in an increased heart rate. So we're affecting cardiac muscle, increased blood pressure, right? So contracting arterial blood vessel walls. So contracting the smooth muscle that surrounds arteries, increasing contractility of the heart. So making heart muscle that is contracting faster also contract more strongly, right? So producing more tension with each contraction, increasing respiratory rate. We start to breathe faster. So the the skeletal muscles that are involved in respiration, and especially the diaphragm muscle involved in respiration, right, is increasing its contraction relaxation rate. It's also increasing the the contractility of those muscles. So we get a deeper contraction and relaxation, which increases respiratory depth, or the amount of air we're able to pull into our lungs, um, via a different sort of mechanism, not acetylcholine release at skeletal muscle, which we expect in the somatic nervous system. It works to increase skeletal muscle tone. So it brings muscles closer to a state of firing action potentials to contract. In other words, it makes it easier for action potentials to occur. It also increases the glycogen availability of muscles. So it causes glycogen breakdown in muscle tissue and increases lipolysis, or the breakdown of fats in fat cells, and increase export of those into the bloodstream so that muscles can use them. Okay, so that's all things that you would need to happen, right, if you were going to run away or fight. Parasympathetic stimulation is alternatively the version or portion of the nervous system that is stimulated when we're relaxed and when we need to take care of basic maintenance activities. This is called often the rest and recuperate or rest and relax portion of the nervous system. So we get the relaxation of ciliary muscles. So now our lens becomes very thin, and it's just sort of like, if you imagine when you're resting your eyes and just kind of staring into the distance, right? Your lens is very thin. And of course, when you're doing that, you're not very attentive, right? You're just kind of staring off into space. Um, Also, your iris or your pupil tends to get smaller as your iris muscles relax and make that pupil shrink. You get secretion of digestive gland from digestive glands, right? So this is portion of your nervous system is definitely turned on when you eat, and you start secreting saliva and hydrochloric acid into your stomach and a variety of digestive enzymes. Um, we the brain, the diencephalon, and specifically the hypothalamus, starts secreting hormones to promote nutrient absorption and nutrient use. So you're basically refueling your muscle cells during when the parasympathetic nervous system is stimulated. Um, there's more smooth muscle activity along respiratory passages and digestive passages. So you're working to sort of move food down your digestive tract, right? Rather than letting go of that, you're moving through food down your digestive tract, and the smooth muscle activity uh, in your respiratory passages is typically to contract to reduce the airway, or the diameter of the airway, right? This is, of course, in contrast to what you expect when you need to take in a lot of oxygen or a lot of air to extract oxygen from it when you're needing to run away or whatever, right? So this would be what you'd get in the case of sympathetic stimulation would be relaxation of the muscles of the airway to allow more intake of oxygen and or of air so that we can extract oxygen from it. The urge to defecate and urinate to get rid of whatever waste products we've got comes with parasympathetic stimulation. And of course, then you get decreases in heart rate, decreases in respiratory rate and respiratory depth, decreases in blood pressure, decreases in the strength of contraction of the heart or of heart contractility. So these are all effects of stimulation of the parasympathetic nervous system. So do notice that just because we're talking about stimulation, it doesn't necessarily mean excitation, right? In a lot of cases, stimulation, right, of the parasympathetic results in relaxation, right? Results in a reduction in metabolism. So just keep that in mind that this is not all about inhibition and this is not all about excitation. Just wanted to bring up our cranial nerves again to before we look at the anatomy of the divisions of these two nerve portions of the autonomic nervous system to just see which one ends up being one that we've talked about already and tested in lab and involved in parasympathetic and sympathetic stimulation, and that's the oculomotor nerve, right? Cranial nerve number 3 here. So we're going to see how that oculomotor nerve is involved. So here's our two anatomical divisions. There isn't much that I need you to take away from this, except that when we compare them, we see that in general, now here is the sympathetic and here's the parasympathetic. In general, both divisions of the nervous system send neurons out to all of the major organs and tissues, right? So we've got the eye, the salivary glands, heart, lungs, the glands and tissues of the digestive system, like the liver and gallbladder and stomach and pancreas, and then of the reproductive system and of the excretory system, both the urinary system as well as part of the end of the digestive tube, right? So that's in comparison. In contrast, you'll see that all of the nerves that are controlling or that are responsible for stimulating uh, these tissues via sympathetic fibers are originating in the thoracic vertebrae, a couple lumbar as well, but they're basically in the thoracic segments of the spinal cord, right? Whereas all these parasympathetic are controlled by the cranial nerves and a few sacral nerves, right? So it's all located in the cranial nerves or a few sacral segments of the spinal cord, right? And of course, these are just for the lower body. But most of this is taken care of by the vagus nerve or the vagrant nerve, the wandering nerve, right? Serves all of these tissues with parasympathetic fibers. Right? Okay. And I just brought up the eye here because here you can see cranial nerve number 3 involved in controlling what's going on in the eye, right? Okay. So I want to take a closer look at these synapses to help us understand what neurotransmitters are being used and in general the effect of those neurotransmitters. Sometimes we can detect a simple effect of these neurotransmitters and in other times not so much. So let's look here at the sympathetic nervous system. You see the preganglionic neurons in the central nervous system. Um, these, and in both cases for sympathetic and parasympathetic, right? So these are having their origins in spinal cord, right? All of these release at their synapses acetylcholine, right? So that means all of these synapses are cholinergic, and all of these synapses here and here are excitatory. So acetylcholine is operating in the ways that you've seen it operate before with skeletal muscle, right? It's stimulating the opening of sodium or calcium channels, perhaps. Right? Now, most of the postganglionic fibers, so here in the sympathetic nervous system, are adrenergic. That means most of them release norepinephrine or epinephrine. And you can see here that um, sometimes epinephrine is released into the blood, but more frequently norepinephrine, this green, is released into the blood. When these cells synapse on targets, they're releasing norepinephrine. So this is the most common neurotransmitter of the sympathetic nervous system, but epinephrine is sometimes also used. So there's two ways. You can either release neurotransmitter directly onto target cells or release it into the blood, right? And then it gets to target cells later, right? So just in terms of thinking about the activity of these, this this activity is very fast because it's acetylcholine, right? So it's extremely rapid, just like skeletal muscle activity. It's over in a couple milliseconds, right? But this activity, because it's released into the bloodstream, and because the way that these actually connect, the way that these postganglionic fibers connect to target cells is not via synapses, it's via a slightly different mechanism. The result is the same. Neurotransmitter is released from this cell. It binds to receptors on the target cell and produces some effect. But we don't have things like, well, we don't have molecules released that digest or get rid of this neurotransmitter very quickly. So these tend to persist for quite a long time, and their activity can persist on target cells for upwards of like 30 minutes, right? That's kind of the outside boundary. So anywhere from minutes to about a half hour, right? So different level of stimulation. Um, If we look at the parasympathetic nervous system, now all of these synapses, pre and postganglionic, are acetylcholine, mediated by acetylcholine. But again, here, these can be longer lasting because they don't have this direct synapse, the way that we saw in skeletal muscle cells. Um, they do release neurotransmitter. These neurons do release neurotransmitter. They do bind to their target cells, but not quite in this nice clean synapse. And so again, we don't have the release of like acetylcholine esterase here to digest this acetylcholine. So these effects can persist for a bit longer too. Okay. Well, let's look at what are these receptors that we have that these neurotransmitters are binding to. We can say a few strict things about how target cells will respond when neurotransmitter is released onto them. Now, the first thing to say is that all these, so these are adrenergic synapses we're looking at. So let me flash back. So we're looking here, right, at the sympathetic nervous system now. All these adrenergic synapses release either adrenaline or oh, sorry, I haven't said this before. Epinephrine or norepinephrine, those are the hormones that are produced, the neurotransmitters that are produced by the body. The synthetic production of those we call adrenaline and noradrenaline, but they're the same thing. They're the same molecule. They have the same effects, basically. So when you hear adrenaline, think epinephrine. When you hear noradrenaline, think norepinephrine. They're basically the same thing. Right? So these bind to G protein coupled receptors, and we saw before that these don't have direct effects in the sense that they are not channels that open when this neurotransmitter binds to them. They have indirect effects, right? They kick off intracellular pathways which alter metabolic activity of the cell or can lead to the opening of channels, but in an indirect fashion, right? It's not directly binding to this and this thing is opening. Right? So here's our receptor types. We've got alpha 1, alpha 2, and a variety of beta receptors, beta 1, 2, 3 receptors. And there are really more than this, but these are the most common and the ones we know the most about. So it turns out that alpha 1 receptors are all excitatory, or when epinephrine or norepinephrine binds to these, they always initiate excitation of that cell. And these are really common, and most of the things that most of the cells that express alpha-1 receptors are smooth muscle cells. So this one is easy. When you get binding to alpha-1 receptors of the sympathetic nervous system, you always get, this is almost always smooth muscle, and you always get smooth muscle contraction. Right? So you get an increase in calcium influx and you get smooth muscle contraction. Alpha 2 are nearly almost inhibitory, and so we're just going to say that they're all inhibitory. Um, when the neurotransmitter binds to this receptor, it ends these pathways, or it inhibits these pathways. So we get the inhibition of neurotransmitter release. We get the we get the inhibition, right? We get the inhibition of smooth muscle contraction. And so again, a lot of these are on smooth muscle, but some of them are on other target cells. And so in general, we get inhibition of that metabolic activity of those cells. Right? Beta receptors are more varied, but for example, beta 1 receptors, when these neurotransmitters bind to beta 1 receptors, these are excitatory. So for example, some of these, and I mentioned this earlier, in terms of making skeletal muscle more able or more ready to use um, uh, nutrient reserves, some fibers synapse on skeletal muscle, and those muscles express beta 1 receptors, and these receptors increase skeletal muscle metabolism, and that means they're just increasing the rates at which skeletal muscle is making glucose available via digesting glycogen, or making fats available via importing fatty acids. They also have the heart muscle, cardiac muscle, expressing this receptor, and so that excites the heart muscle and increases the heart rate and increases contractility, or the strength at which the heart contracts. Right? Whoops. Um, Beta 2, these are generally inhibitory receptors. So when these neurotransmitters bind to a beta 2 receptor, they tend to inhibit and this is mostly on smooth muscle, right? They tend to inhibit smooth muscle contraction. So the smooth muscle dilates, right? So smooth muscle relaxation. And then finally, beta 3 receptors, we tend to see these on fat cells, and these are generally excitatory. And so when we get this norepinephrine or epinephrine binding to beta receptors, we get an increase in metabolic rate, an increase in the liberation of fats via lipolysis, so increase in fatty acid breakdown and export into the bloodstream. And if you think about it, all these are just in keeping with what we said we need to do when we excite the sympathetic nervous system. Make more nutrients available, make the heartbeat faster, make it beat more strongly, right? Um, uh, Cut off, for example, the blood flow to the stomach and the digestive tube, increase, right, blood flow to the skeletal muscles, right? So all of these kind of are in keeping with what we said should happen when we stimulate the sympathetic nervous system. Um, well, let's take a little more a closer look at a couple of other receptors. Now, these are you know that these are, because we have acetylcholine here, right? These receptors are in both the sympathetic and parasympathetic nervous system. But when we're thinking about effects on target organs, right, we're really talking about the parasympathetic nervous system. Let me flip back just to remind you, right? We have acetylcholine released here onto these cells, but when we're talking about effects on target organs or target cells, we're really thinking about the parasympathetic nervous system, because that's where acetylcholine is released. So those receptors are of a couple different types. We have, and there are subtypes too. There are nicotinic receptors. These are stimulated really strongly by nicotine, and they're abbreviated with this NACHR. So nicotinic acetylcholine receptors. And we have muscarinic receptors. Muscarine is a toxin originally, or a compound originally isolated from a mushroom species, and a whole family of mushroom species produces these this muscarine. So these are abbreviated muscarinic acetylcholine receptors. Okay, well, what do these do? Where do we find these? Well, we know we find these these acetylcholine receptors on all the preganglionic nuclei of the autonomic nervous system. So all the parasympathetic and sympathetic preganglionic nuclei, right? We just looked at that. So all of these are nicotinic receptors, right? All of these are nicotinic. And we also, of course, find these on all the neuromuscular junctions, neuromuscular, skeletal neuromuscular junctions, right? We saw that when we looked at the muscle system. So they're pretty widespread. These are easy. When these are stimulated, so when acetylcholine is released and binds to this receptor, a nicotinic acetylcholine receptor, it always excites the postsynaptic cell by opening sodium channels. So let's just flip back one more time, right? When acetylcholine is released here or here, it always excites, right? Always excites. And most frequently, it's on these sympathetic cells or sympathetic neurons. It always excites these postsynaptic cells. Right? And the muscarinic receptors, they're on all the parasympathetic and a few cholinergic sympathetic junctions. So they're on all of these parasympathetic, right? And a very few of these, but all of these junctions here, right, are muscarinic acetylcholine receptors. So all the parasympathetic. What do? These are G protein coupled, right? So these are G protein coupled, just like we saw epinephrine and norepinephrine were G protein coupled or bound to receptors that were coupled to G proteins. This acetylcholine receptor, this muscarinic acetylcholine receptor, is also coupled to G protein coupled receptors. So again, their effects are longer lasting, and kind of like our beta receptors that we saw in the sympathetic nervous system, these muscarinic acetylcholine receptors can either excite or inhibit their target organ. And they do that by altering their permeability to potassium, right? So either by increasing their the potassium, or sorry, increasing permeability to potassium, right? That's going to inhibit the cell, or decreasing permeability to potassium. That's going to make it easier to bring the cell to threshold and to excite it. Right? Okay, so that's it for our autonomic nervous system. Just a little bit here, and I'll give you some examples, like, oh, what if you had a toxin that bound to and inhibited these alpha 1 receptors? What would you expect to happen, right? So hopefully you'd be able to say that you would have any smooth muscle that expressed this would no longer contract. So it would just dilate, right? So you'd get, and maybe I'd place this for you. Like maybe I'd say the beta 3 receptors, or the beta 2, the beta 1 receptors on heart muscle or on cardiac muscle. So hopefully you'd be able to say that, well, if a toxin or some kind of compound binds to this, right, and is an agonist of this, right, then it's going to increase the activity in this cell. So you're going to get, for example, increase in heart rate and an increase in the strength of contraction of the heart. Right? So think about it in these sorts of terms, right? Or think about, play these little games like, oh, what if I, had some toxin or some drug that suppressed the activity of this receptor or increased the activity of this receptor or whatever. Okay, please let me know if you have any questions about this.